So clozapine the method, we are going to start the clozapine method validation. So clozapine the method validation means first we have to select the pure column that will be obtained from sigma Aldrich. We have already obtained the sigma Aldrich pure column. Then we have to select which column is feasible for the clozapine method validation. उसमें I have selected the CAP column will be better, but उसमें extra double bond column will be better for the clozapine method validation because clozapine is a lipophilic compound. So if you are using normal RP column, C18 column, reverse phase column, or docile column is there. If you are using, some chance of retention of the compound itself, the column will be there, so that it can cause a variability in the retention time. मैंने आपको दिखाया था retention time. Retention time mainly एक fixed retention time, fixed experimental condition में आना चाहिए. But it can vary sometimes in column का affinity की वजह से it can vary in multiple runs. Variability can occur, so that can be avoided if you choose the correct column. In clozapine method validation, we are selecting the column C80 reverse phase only, but extra double bond XDB column I am selecting. Extra double bond column I am selecting for the clozapine method validation. So column तो select हो गया, pure compound भी मिल गया. Pure compound we obtained from sigma Aldrich. Then we made the standard of the clozapine, so that the concentration from higher concentration to low concentration, and one concentration above the therapeutic level, one concentration below the therapeutic level, 3-4 concentration through the therapeutic level, Then this below the therapeutic level is for LOQ and LOD determination. LOQ is limit of quantification and LOD is limit of detection. And उसके बाद हमने रन किया, chromatogram मिला. We have explained about the chromatogram also and इसका properties also. उसके बाद chromatogram से Excel MAU निकाल के, concentration निकाल के भी Excel में feed करके linearity check किया. It is R square is also good done. Then we have to, once the standard the whole thing is made, then we have to extract the compound. अब मैंने patient को clozapine दिया, clozapine देने के बाद 2 hour बाद oral tablet है, so absorption के लिए time लगेंगे. Oral tablet है तो 2 to 3 hours के बाद मैंने T concentration देखने के लिए blood निकाला. That is an EDTA bottle or या plain bottle. Plain bottle means pink yellow colored vial. EDTA means pink colored vial. And after the I have taken this EDTA bottle में blood लिया मैंने. उसको मैंने then I immediately EDTA bottle में या pink bottle में yellow bottle में लिया हुआ blood को centrifuge किया. कितने में centrifuge करना है? You have to centrifuge at 4000 RPM for 4 minutes. So it will be, you will get the vial like this. You will get a clear fluid above. This is clear one, and this is the pelleted portion. So this is a plasma or serum. You can get plasma or serum. Then we will shift the plasma or serum to another append tube. Clear append tube, plasma or serum shifted. And this can be immediately tested. Yeah, it can be stored a long time in -80 or -20 degree for estimation. If the immediately testing is not practicable or feasible, we will store this in -20 deep freezer or -80 degree deep freezer for long time storage. Clear? Once you started the method, you are going to estimate from the plasma what are the components can occur inside the plasma. Plasma में तो sugar protein होगा, albumin होगा, globulin होगा, other compounds होगा. So you need not do these things because that should be extracted from the plasma. Plasma should be cleared of this high molecular weight compounds like proteins, amino acids, and that amino acid नहीं है, proteins mainly high kilodalton compounds should be extracted out from the plasma. So that process is called a plasma extraction. Clear? So this plasma extraction is a process by which we get only the solvent or solution dissolved with the now desired compound to be detected. Clozapine है तो clozapine या phenytoin है तो phenytoin या metoprolol है तो metoprolol उसमें dissolve होगा उस solvent के अंदर. उससे करने के लिए हमें 200 microliter of plasma लेके उसमें 300 microliter of acetonitrile compound add करना है. Immediately after adding, we will keep it for vortex for 1 minute, and after that we will keep it for 30 seconds. 30 seconds उसमें ऐसा ही रखेंगे. उसके बाद we will vortex करने के बाद it will some of the compound will sediment out and some clear fluid will be there. But after that we will centrifuge the same Eppendorf tube. Centrifuge it for 14,000 RPM for 5 minutes initially. Then we will have a get a compound like this. Some pellet will be there, some clear fluid will be there now. Then we will transfer this clear fluid to another Eppendorf tube without pellet. Then again centrifuge the same thing for 14,000 RPM for 5 minutes. Clear? For 2 minutes. 5 minutes, 2 minutes. उसके बाद we will get the very pure liquid only containing the drug dissolved. समझा है ना? So this entire process of extraction of the compound dissolved in the solvent is called a solid liquid extraction. Why it is called a solid liquid extraction? So we will convert the plasma into two interfaces. One is pelleted part, that is solid, another is solvent, pure solvent, that is liquid. Again we will transfer out and liquid, and again pelleted part, again we will transfer out, then only this liquid which is dissolved in the drug. So it is called a solid liquid extraction. For the solid liquid extraction, which solvent we are using commonly? Acetonitrile. And methanol can also be used, but acetonitrile is the common compound used for protein precipitation. So this entire process is called solid liquid extraction. Some compound solid liquid extraction is not practicable, so we can use another method of plasma extraction, that is called a liquid extraction. So what the difference between solid liquid extraction and liquid extraction is that instead of acetonitrile we will use either diethyl ether or ethyl acetate. So ethyl acetate or diethyl ether. We can use ethyl acetate or diethyl ether for liquid extraction. So the step is only changes, you have to use 2 times or 3 times of ethyl. So 500 microliter diethyl ether or 500 microliter ethyl acetate we can use. Then we will get what compound. The first procedure same. vortex करना, 30 second रखना, centrifuge करना. This centrifuge करने का step के बाद you will get a like this one, 2 layer of solvent liquid. One heavy liquid, नीचे heavy liquid होगा, ऊपर light liquid होगा. उसके बाद this should be kept in -20 deep freezer for कितना time? 2 minutes. So it will be solidified. The नीचे वाला part will solidify, ऊपर वाला part will still containing the solvent with the drug, that is undissolved. Means it will become liquid only. Then we will take the liquid part from the top and we will centrifuge that one more time 2 minutes, and then we will take load into vial for run of the Hplc. So this entire process of extraction is called a liquid extraction. Why it is called as liquid extraction?